Namaste to all students. Khushbu myself JV'n Dr. Khushbu Verma, Faculty of Agriculture and Veterinary Science. Today we will discuss about role of cyclins and CDK in cell division. And this session is related to course Cell Biology of program B.Tech FBT, B.Tech M.Tech FBT and B.Sc FN&D third trimester. So we will discuss here about the roles of cyclins and CDK, cyclin dependent kinase. So we will look at a few of the most important core cell cycle regulators like proteins such as cyclins and enzymes called CDK means cyclin dependent kinase, and an enzyme complex is called APC/C. First of all, we will discuss about the cyclins. So, cyclins are among the most important core cell cycle regulators. Cyclins are a group of related proteins, and there are four basic types found in humans and most other eukaryotes. First is G1 cyclin, next is G1 oblique S cyclin, then S cyclin, and last one is M cyclin. As the name suggests, each cyclin is associated with a particular phase, transition, or set of phases in the cell cycle, helps derive the events of that phase of or period. For instance, M cycling promotes the events of M phase such as nuclear envelope breakdown and chromosome condensation. Here we can see this graph where G1 cycling concentration is getting high at a particular time period, then rapidly decline when M phase is completing. And G1 oblique S cyclin is at its high peak at S phase, and S cyclin is highest at G2 phase, and M cyclin is highest at the M phase. So levels of different cyclins vary considerably across the cell cycle, as shown in the diagram at the top. And a typical cyclin is present at low level for most of the cycle, but increases strongly at the stage where it's needed. M cyclin, for example, peaks dramatically at the transition from G2 to M phase. G1 cyclins are unusual in that they are needed for much of the cell cycle. Now we will discuss about CDK, cyclin dependent kinase. In order to drive the cell cycle forward, a cyclin must activate or inactivate many target proteins inside the cells. Cyclin derive the event of cell cycle by partnering with a family of enzyme called CDK. Alone CDK is, alone CDK is inactive, but the binding of a cyclin activates it and making it a functional enzyme and allowing it to modify target proteins. How does it work? So CDK are kinases enzymes, and that phosphorylate means attach phosphate groups to specific target proteins. The attached phosphate group acts like a switch, making the target protein more or less active. When a cyclin attached to CDK, it has two important effects. It activates the CDK as a kinase, but it also directs the CDK to a specific set of target proteins once appropriate to the cell cycle period controlled by the cyclin. For instance, G1 oblique S cyclin sends CDK to S phase targets, examples like promoting DNA replication, while M cyclin sends CDK to M phase target means for making the nuclear membrane breakdown. Here we can see if there is no kind of a cyclin, so CDK will inactive and it will not bind or it will not direct to the target any of targets, and S phase factor will always off. But G1 oblique S cyclin, if present over there, so it will attach to the CDK, and then CDK will be active and it will go to the target 1, 2, 3, and it phosphorylate to them, and Then S phase begins, means DNA replication enzymes are activated and replication process further will be started. Now, maturation promoting factor, a famous example of how cyclin and CDK works together to control cell cycle transitions is that of maturation promoting factor MPF. The name dates back to the 1970s when the researchers found that cell in M phase contained an unknown factor that could force frog egg cell to enter M phase. This mystery molecule called MPF was discovered in 1980 to be a CDK bound to its M cyclin partner. Here we can see in the diagram, like if there is M cyclin, so it will be attached to CDK, then CDK is activated, it will phosphorylate its targets, and M phase begins, means spindle forms, chromosome condensation will be start, nuclear membrane will break down. Example, nuclear membrane breakdown. Here we can see nuclear membranes are properly present, but when CDK will bind, to the M cyclin, so it adds phosphate group the targets and membrane will start to break down or will to be disappear. Next one factor is anaphase promoting complex or cyclosome, which means APC oblique C. In addition to driving the events of M phase, MPF also triggers its own destruction by activating the anaphase promoting complex, a protein complex that causes M cyclin to destroyed. Starting in anaphase, the destruction of M cyclin pushes the cell out of mitosis, allowing the new daughter cells to enter G1. The APC oblique C also causes destruction of the proteins that holds the sister chromatids together, allowing them to separate in anaphase and move to opposite poles of the cell. Here we can see in the diagram, APC complex will add ubiquinin to the saccharin. Here, in general terms, um, when chromosomes are not going to divide in NFA, or you can say cell is not across the metaphase stage, so saccharin will be attached to the separase, and this combination is inactive. Means separase will be inactive when saccharin is attached to it. But anaphase promoting complex will add ubiquinin to saccharin, and this saccharin now it's this modified saccharin will be separated from the separase and it will be breakdown, and now separase is free and now it is in active form, so it will break down the chromosome into two sister chromatids, and this is cohesin, which requires two binding two sister chromatids with each other. So it will break down or it will chop up the cohesin, and sister chromatids will be separated further for anaphase stage. So this is the sum up of different regulations of M cyclin, different regulations of cyclin and CDK. And this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University. Hope you are satisfied with this session. If you have any query, then leave comment and I will resolve it. Thank you.